Hello everyone, my name is Madison Pollard. I am a Broward College bachelor student studying physical environmental science with my main focuses being in oceanography and benthic ecology. The Everglades Protection Area was known to historically support 70 to 90% of nests annually. Since the early 1900s, humans have compartmentalized the natural hydrologic flow of freshwater within Everglades National Park into storm treatment areas, water conservation areas, canals, and levees to acquire farmland and residential space. Degradation has since occurred and has precipitated changes in the number of successful colonies in both tactile and visual feeding wading birds. Due to their hydrologic prey foraging patterns, they rely on the quality, quantity, timing, and distribution of water flow. Usual habits like timing of nesting and reproduction have been observed to be influenced by high and low quality wetland areas, where prey vulnerability and concentration can vary. The purpose of this project is to study water levels, proximity to human activity, the colony longevity, and these factors' ability to influence restoration of wading bird nesting colonization in Florida Everglades National Park. Sy and their colleagues examined 40 years of annual aerial surveying data of wood storks to determine the relative importance of colony variables to colony dynamics. Colony site attributes were classified as active versus non-active, whether there was presence of human disturbance within 500 meters of a nest, continuous versus discontinuous, and the local hydrology was recorded for each site. Evidence of human disturbance included roadways, recreational activities, agriculture, and logging. Lorenz tested the relationships between water level, food availability, and nesting success of the roseate spoonbill by prey fish sampling, collecting water level data, and examining nesting colony surveys. Fish collections occurred during transitional and dry seasons using drop traps and subhabitats at each site. Water level data was determined by recorders placed in known foraging locations. Nesting colony production was determined by visiting the sites on a 7 to 10 day cycle, and if a nestling was observed to survive for 21 days, the nest was considered successful. A mean water level, prey density availability index, and prey concentration threshold was analyzed during the 21-day nestling period. With Sy and the wood storks, colonies were observed to be influenced by size and longevity. Colonies near the mainland did not persist, but colonies closer to a limited degree of human activity, for example, water conservation areas, had higher probability of recolonizing the following year. With Lorenz and the roseate spoonbill, the prey concentration threshold was determined to be 13.15 centimeters. Lorenz found that there was not a linear relationship between water level and prey concentration. Lorenz also determined that an individual nest could produce up to 4 chicks at lower water levels compared to failed nesting at levels above the PCT. Nests that produced more than 1 chick were present in areas of higher density availability indexes, rather than those that produced an average of 0 chicks in areas of low DAI. To conclude, restoration of wading bird nesting is influenced by water levels, proximity to human activity, and site longevity. Based on Lorenz and Sy's data, using wading birds such as the wood stork and roseate spoonbill as ecological indicators can aid restoration managers in adaptive management practices. An increase in populations near water conservation areas hint at the need for some human intervention. I would like to thank Dr. Pamela Fletcher at Broward College for the time she's always dedicated to helping me expand my knowledge of restoration. Also, thank you to the Broward College librarian, Elena Hall, for helping me with my citations. Last but not least, thank you Dr. Antonia Florio, the Science Communication Liaison for the National Park Service, for guiding me and giving me pointers on my resources.